All right, lecture 20.2. We're going to talk about heart excitation and the conducting system. So basically, what are the electrical and contractile components to our heartbeat? So when we talk about a heartbeat, gunk or ba-bump, that is one cardiac contraction. So it comes in a couple of parts. We're going to break that down and take a look at it on an EKG or an ECG. And basically what happens is the upper chambers contract first. So your atria have an atrial depolarization event, AKA a contraction. And then the ventricles have the same. And while the other set of chambers are contracting, the other group is relaxing, and then there's a brief period where everybody's in relaxation. There are two types of cardiac muscle cells that we have. The first are called autorhythmic cells, and basically these are the cells that initiate and control and coordinate the heartbeat, because that is internally controlled within the heart muscle. And then you have contractile cells. So you have cells that are actually going to do the heavy lifting, if you will, actually do the contractions that result in blood being propelled throughout the body. So what we're going to look at is that conducting system today, and that's going to focus on a few different cell types within the heart itself that are going to, as I mentioned before, initiate and spread that electrical impulse to initiate contraction in the cardiac muscle. And what's cool about the heart is that it's autorhythmic, all right, or it has autorhythmicity. And basically what that means is that the cardiac muscle tissue does not need neural stimulation to initiate contraction. That's why someone can be brain dead, if you will, but still have a heartbeat. And that's where we get into kind of a very gray area of, when is someone alive? When is someone no longer alive? So very interesting science here related to the heart, that it has this kind of own self-initiating part to it. So there's a few different components that we're going to be looking at. So you can see the two primary groups. The first group are the pacemaker cells. So you have two nodes that initiate and control that pacemaking for the heart, and that is the SA and the AV node, or the sinoatrial and the atrioventricular nodes. And then you have the conducting cells. So you basically have these pathways both within the atria and the ventricles that are going to spread that impulse to all of the contractile cells to result in both atrial and ventricular contraction, right? In terms of what happens, there is, as controlled by the SA node or the pacemaker itself, establishes what we commonly refer to as the sinus rhythm. And basically that's what you see, depicted on those kind of EKGs or ECG images where you see the heartbeat, visually shown on those electrocardiograms, and allows us to see what that normal rhythm would be when we have proper contraction and initiation by the SA node. So here's an overview of what those components are. And I believe you have this to label in your packet. So you have two nodes. There's the first within the right atria, and that's the SA node, commonly referred to as the pacemaker. So that's going to be our initiating conduction portion of the conduction system, And then we have the second node, and that is the AV, or the atrioventricular node, located right between the right atria and the right ventricle. And so that electrical impulse is going to go from node to node. There's a little bit of a delay happening in there we'll talk about. And then we're going to conduct that. So we're going to send that electrical impulse message through the AV bundle. And then it's going to branch into right and left bundle branches. And then eventually we're going to get to kind of where we see this branching off, and those are referred to as the Purkinje fibers. And that is where that electrical impulse is going to be stimulating those contractile cells within the ventricles themselves to allow for ventricular depolarization, AKA ventricular contraction. So here's kind of a breakdown, and what's nice about these images is that you see the ECG, or the electrocardiogram images, because we are going to focus on this shortly. So it kind of gives you a little highlight of where on the EKG or where on the ECG you would be seeing this activity. So first of all, there is that pacemaker or SA node initiation, and usually that happens quite frequently. So an action potential is simply kind of a nervous impulse initiation. We'll talk more in detail about that in April and May. but usually you have anywhere between 60 and 100 of those initiating signals coming from the SA node. Following that, what we see on the ECG is a P wave, so it's that small wave here in red. And basically what happens is those internodal pathways through the atria are going to spread that initiating pacemaking electrical impulse from the SA node to the AV node, located between that right atria and right ventricle. And what we see with that P wave and why there's a little bump is that we have what's called atrial depolarization. Depolarization is a nervous system term, and since we haven't covered that yet, for us, we're going to equate depolarization with contraction. So anytime there is a depolarization event, that means that we have initiated a contraction. And in this case, the atria, both the right and the left atria, are going to contract and squeeze out any remaining blood into the ventricles. Then there's going to be a pause. So at the AV node, it kind of resets the race, if you will. And that's what you see in that PR interval, where we have the delay of that impulse, because we have to get that impulse from the node through into the bundle branch. We do that by going through the AV bundle. So going through these fibers that are going to head into the interventricular septum. So we can make our way down into the ventricles to result in a ventricular contraction. So we've got about, I think it's 100 or so millisecond delay before we head into the conduction portion. All right. So once that happens, we are going to hit what's called the Q, this will be R, and then S complex. And so Q is going to be our initiating of ventricular depolarization, aka ventricular contraction. And that happens because we are now sending out that impulse through the AV bundle into the bundle branches and heading towards the Purkinje fibers. And what's that going to start to do is it's going to initiate the papillary muscles. It's going to stimulate those papillary muscles to be able to close up those AV valves. And we want them closed so that we can contract the ventricles and that our blood flow can go in one direction, up and out, either through the pulmonary semilunar valve or through the aortic semilunar valve, and go either out to the lungs or out to the aorta and the rest of the body. All right, so during that, again, QRS complex, ventricular depolarization happens. So that initial Q depolarization allowed the papillary muscles to close up the AV valves, kind of like pulling a drawstring, bag closed. And then the rest of that impulse can spread through these Purkinje fibers into the myocardium of both the right and the left ventricle. And then that blood can get pushed up and out through the pulmonary trunk or through the aorta. And that pushes all that blood out, does what the heart needs to do. All right. Sometimes, though, that conduction system, so that initiating of that SA pacemaking node impulse, the delay at the AV node, the spreading through the conduction system, sometimes that doesn't work right. And here are a couple general terms. You'll be doing an activity that'll talk about some more specific ones. But there are two general terms for heart rhythm irregularities or arrhythmias, and bradycardia or tachycardia, or being bradycardic or tachycardic. When someone has bradycardia or they are bradycardic, it means that their heart rate is slow. So some initiating factor within that S node is failing. And so they are not regulating the proper, 60 to 100 impulses per minute. If someone is tachycardic, that means that their heart rate is abnormally fast. So it's initiating that impulse way too quickly. So way more than 100 times per minute. And so that obviously can cause some issues with the rest of the conduction cycle, your blood flow, oxygenation by the time they are getting the blood to the lungs for oxygenation, and then back to the body as well. We're able to see and read this by looking at what's referred to as an electrocardiogram, abbreviated in English as ECG, but you more commonly hear it referred to as an EKG in German, which is where this term originated. Cardio is spelled with a K. So either term is fine. You'll hear me try and use ECG more often, but basically it records the electrical events in the heart. Typically any doctor show, medical show, you'll see someone hooked up to an EKG where you can see, the visualization of these impulses and this conduction system. And basically what needs to happen is they need these electrodes placed around the heart so that they can see. And usually this is something done to track people, especially, post-surgical or pre-surgical, And then obviously if you have any conditions that would result in some type of abnormality or arrhythmia of the heart. And so what you're reading on the ECG are those waves or complexes that I mentioned before, the P wave, the QRS complex, and then one last piece, which is the T wave. All right. And generally speaking, this is important. So make sure you're making note of this on your packets. The P wave, or that first wave, is when we depolarize the atria, AKA the two atria contract. All right. So they're going to squeeze out blood into the ventricles. Then we're going to have that QRS complex, so the big peak that you see. And what that consists of the ventricles contracting, both right and left. So that is ventricular depolarization. And again, that starts with the closing of the AV valves, right? Or not starts, but a part of that is closing of the AV valves by that electrical impulse triggering the papillary muscles to close up those drawstrings. And then the actual contracting of the two ventricles to push that blood up and out, either to the lungs or to the rest of the body. After that ventricular contraction, we see a little T wave. And basically, what is happening there is we have repolarization. And repolarization is of everybody, not just the ventricles, but the atria are also relaxing. And repolarization, we're going to think relaxation. So basically, all four of the chambers take like a moment to breathe, if you will, and relax. But we do see a T wave on our EKG because what's happening is those AV valves are closing, and we have kind of a bump, usually in the aorta, of that blood flow coming back. Remember when we looked at the AV valves, they have those little cusps, when you push open the saloon doors and they fold back. All right, that blood is going to kind of pool in there, and that creates a little bit of pressure right in the aorta, right outside the aortic semilunar valve. And so we end up with this little bump on the ECG, which we record as the T wave. And that just indicates for us that everybody's in relaxation. We closed up the semilunar valves, pulmonary and aortic, and everybody is just kind of chilling out. All right. So this is really important that you can talk yourself through the ECG reading so you know what a normal one looks like. You're going to have a chance to look at some abnormal ones. So sometimes the spacing, all right, so you can see time here on your x-axis. Sometimes the spacing of PQRST on an ECG is going to be closer together. That would indicate some type of tachycardia, right? We're going too fast. Sometimes those segments are going to be spread out further, and that would again indicate bradycardia, so a slower heartbeat. And then you're going to see pretty much everything in between. Sometimes your P wave is going to be absent. Sometimes the QRS complex is going to be askew. Sometimes you're not going to have a proper T wave. So anytime you have an irregularity in one of those segments, it indicates to you something wrong with that part of the conducting system. And that's why it's important to read these ECGs and to be able to interpret them as well. The next couple of slides go through some of those irregular ECG readings. So you can just see right now with just these three here, variations significantly from that normal looking ECG reading. So you can have, these are called PACs or PACs, premature atrial constriction. So the atria are contracting too much. So you're initiating like 2 or 3 impulses here. You could have atrial tachycardia. So the atria are contracting way too quickly to keep up with the ventricles. You could be in AFib, all right, or atrial fibrillation. And that, again, you can see with the numerous amount of atrial contractions, and your ventricles can't keep up. You could have PVCs, or premature ventricular contraction. So your ventricles are contracting too early, all right, without waiting for that electrical impulse process in the conduction system. And then you can have ventricular tachycardia or VFib, ventricular fibrillation. And because these are all controlled via electrical impulses, that is the reasons why we can use defibrillators or those paddles, right? Rub them together, clear. We can use electricity to be able to try and reset the heart because it is electrically controlled within the heart tissue itself. Last piece here for conducting system has to do with the contractile cells. And in those cardiac contractile cells, there is a structure called an intercalated disc. And basically, the membranes within the cardiac muscle cells are linked together by what's called a gap junction. And basically, that is a little bit of space, which allows for the electrical impulse to jump from one cell to another. And what happens is that these intercalated discs, all right, we can see an example of them in here. And these intercalated discs basically allow, let me give a good one, here we go. So we can see these intercalated discs. We're looking at a SEM of, or a light microscope version of cardiac muscle. And basically these intercalated discs provide the opportunity for those electrical impulses to kind of move faster. So think about if you were trying to get across like a pond or something. If it had little stones to take steps and hops through, you could probably hop on each one of those faster than you could swim the pond. And that's basically how an intercalated disc works. It allows for the electrical impulse within the cardiac muscle to be carried faster. And that is what allows for the heart rate and that electrical impulse and atrial and ventricular contraction to happen as fast as it does.